So welcome to the CTSA Precision Medicine Resource Seminar Series. I'm Gary Miller. I'm one of the co-directors of this resource. And it's my pleasure to introduce Dr. Gary Patti from WashU here. Uh, as you'll hear, he does a lot of great work with mass spectrometry and metabolomics and has really been a pioneer in this area. Every sort of chemistry award from Malincrot, Beeman Medal, Sloan. He's like won all of those things you can do throughout your career. He's been incredibly innovative, including starting a company building off a lot of this technology. And he was also a recipient of one of the National Institutes of Environmental Health Sciences River Awards, which it's kind of like NIH's version of like a Howard Hughes thing, where you give people multiple years of freedom to do innovative things. And I think that he was an excellent choice for that because, as you'll see, what he's been doing is just really having a great impact on the field. So we are thrilled to have him here to talk about the work he's doing in this multi-omic stuff, and take it away.
Thank you so much. Yeah, thanks for the introduction, and it's really a pleasure to be here today. I'm going to talk about the application of multi-omics in the context of precision medicine. And I know that's the idea of this seminar series, and I hope to paint a good picture of what that looks like and how I hope that we can contribute. My talk today is going to contain 4 parts. First, I'm going to talk a little bit about the motivation for doing multi-omic studies. Then I'm going to talk about how the success we've had at the population level as we build up to do large-scale studies. Then I'm going to tell you a story about the intersection of the diet and specifically the environment, fructose, and cancer. And lastly, we'll talk about some implications in the context of human disease. The wrong button there. Okay, so we'll start with the first topic here, motivation for multi-omics. So this is a diagram that I got from Jill Moore at UMass that depicts the vision. One way of it's an analogy for how you might think about doing multi-omics. And I really like this. This is it equivocates multi-omics to building a house, where each of the ohms plays a different role in the construction of the house. So you can imagine that the genome is the blueprint for the house, which of course you need to build the house, but you also have to abide by all the zoning codes. Can you have a deck? Where are the property lines? All those sorts of things. And you can think of that as being the epigenome. The transcriptome is really the building materials, the 2x4s, the bricks, everything that you need to establish the house. Then above that, you have the proteome and the metabolome. You might think of the proteome as being the construction workers, the enzymes that come in and do their diligence. The metabolome and the lipidome, you might think of as being the furnishings for the house. And then lastly, at the top, perhaps among the most important, is the exposome. And you might consider that to be the weather that the house has to endure and how that weather influences the structure of that entity. I really like this metaphor, as I said, but I'm going to show you another metaphor, if that didn't work for you, that is going to relate maybe something a little more familiar, American football. So this is arguably the greatest catch in Super Bowl history. I realize I'm presenting this in New York to people who maybe aren't so enthusiastic about Boston. Sports, in terms of sports, this is a Patriots Tom Brady throwing a pass to Julian Edelman, and you watch this play live. And the question that you ask is, did he actually catch the ball? Right? Did he come up with this catch? And it's really difficult to tell when you watch it in real time. However, because it's the NFL and they have ungodly amounts of money, they record every play with 8 different cameras. So you can watch this from different camera angles. And I would say that doing multi-omics is really like watching this play from a different angle. And every play, here, I think you can see, okay, that angle that we're watching now, he maybe he caught it. This angle is not very helpful, I think. You just can't tell because you see his back. This angle, in my opinion, really nails it. This is where you can see, oh yeah, he's got it. It did not hit the ground. This angle, I don't know what it offers. But here, it was the bottom left corner that really told us that he caught the ball. However, in other plays during this exact Super Bowl, other camera angles were the most important. And the reason I take a moment to say that is because I think one thing that happens in multi-omics is that we we argue about which ohm is the best. And you're a genomist, you know, genomics is the best, or epigenomics, or it should be exposures, or whatever it might be. But it really is context, just as this visualization hopefully shows, it really is context dependent. It depends on the play, what camera angle you want to use. And biology is the same way. It's complex, and every ohm gives us a different perspective on how to look at the disease, and exactly which perspective we need is going to depend on the disease and a bunch of other factors as well. Now, what we do in multi-omics is not look at each play individually, but we want to take all of these different ohms and integrate them. And this is really the equivalent of watching all of these plays simultaneously and stitching them together. And so if you're an NFL fan, you'll appreciate that, okay, he caught it, but where was his knee? Well, you can't tell from this angle. I can tell that he caught it from this angle, but I have to look at the other angle to see where his knee is, or to see if his elbow was down, or, you know, all these other aspects. So we want to stitch them all together, and that's really data processing. Now, multi-omics has been around for decades, but it's only been recently that the NIH launched this consortium, which I know we have some participants here. Columbia is a side of this. But this consortium, which is called MODE, which is the Multi-Omics of Health and Disease Consortium. And the structure of this consortium is shown here. This is the red sites there represent coordinating centers. The green sites represent the DSSs, disease sites, or places that are generating the samples. And then there's an omics production center, which we are excited to be leading there at WashU. And I point out that, as I mentioned, Columbia is a center, a clinical center that's led by our colleague Christoph here. So this is the structure of the consortium. And one of the motivations for doing this, building this consortium, was that although, as I say, multi-omics has been going on for a while, many years, there is some sense, depending on who you talk to, that perhaps it has under-delivered and maybe a little underwhelming compared to what you might have hoped. And one possible explanation for that is that there is some disparity between the way in which it's done. So what I mean by that is, what often happens is that there is a group in New York, maybe, who does proteomics on breast cancer. And then maybe there's a group in San Diego who does metabolomics on breast cancer. And then maybe there's a group in Seattle who does transcriptomics. And then there's some informaticist in Chicago who tries to take all these data sets and interpret them together. The problem is that these data sets weren't really designed harmoniously, and they, you know, the ages of the patients might be different. The oncogenes involved in the cancer might be different. The treatment modalities might be different. And so it really is apples and oranges, and trying to integrate them is challenging. And so one major goal of this consortium is to try to address those issues by trying to harmonize everything from not just the way in which we think about which samples to go after, but the way the samples are processed, doing everything on the exact same samples in parallel. Some of the other goals of the consortium are listed here. Of course, we're hoping to expand our knowledge of selected diseases. But two of the other goals, which particularly exciting to me, is that we're hoping to establish best practices for doing multi-omics analysis. So if you're going to take a specimen and you want to do all these ohms on them, how do you do that? I should point out, in this consortium, we're collecting data on 9 different ohms. So that is, let's see if I can name them all: whole genome sequencing, whole genome bisulfite sequencing, ATAC-seq, RNA-seq, proteomics, metabolomics, lipidomics, Metabolomics, and Exposomics. So it's a lot of data that we're going to generate. And the idea is to figure out best practices to try to do these multi-omics analyses that extend, of course, beyond the consortium, and then also to provide exemplary data that everyone in the community can use. So it's harmonized, and hopefully that this will be a source of a resource for others, maybe who are developing computational tools, or who are thinking about other ways to approach multi-omics, can take advantage of. So if we think about what we could use multi-omics data for, I want to refer to a paper that was published in 2021 in Cell by the former director of the NIH, Francis Collins. And that paper was called 7 Ways to Transform Healthcare by 2030. And the idea that is invoked here is that for precision medicine to be effective, we need these three attributes. We need large cohorts. The cohorts have to be diverse. The diversity has to be representative of the community. And the cohorts need to be longitudinal, meaning that we need more than one time point. We can't just collect samples from, a static snapshot of time. We need prospective cohorts. As I said, I think this, if we're going to take multi-omics and we're going to deliver an advanced precision medicine, these are three very important characteristics that we need to consider. And I think we're doing a better job of big and diverse data sets, but I think one aspect that we continue to fall short on, in my opinion, is longitudinal analyses. When I say longitudinal analyses, I don't mean that we want samples over a week time span. I mean that we probably are going to need samples throughout the entire life course of the individual, going all the way back to birth, or perhaps even in utero. And to try to underscore the significance, because I think this is the easiest one to ignore on this list, is the longitudinal. But to underscore its importance, I want to tell you just very briefly in one slide about a recent set of experiments that we did using zebrafish. So we did this experiment where we took zebrafish larvae. These were 5 days post-fertilization, and we exposed them to 1 mg per liter of a compound called BPF, bisphenol F. Now, you may not be as familiar with BPF. This is an endocrine disruptor. It's very similar to BPA. And the reason that BPF, unfortunately, if we surveyed all of your urine here in today and analyzed it, about 80% of us would have BPF in our urine. It's a ubiquitous compound. It's used in plastics and resins. And so we're all exposed to it. It used to be, we used to primarily use BPA, bisphenol A, but there have been, BPA's gotten some bad press, as it should have. And there's been some partial bans instituted on BPA. And so the chemical companies have responded, which brings up a whole nother policy issue, but responded by using BPF, which is simply 2 methyl groups different than BPA. Okay, but what we did is we took these animals that were 5 days post-fertilization larvae and exposed them for a very brief period of time to BPF, just 48 hours. Then we waited 6 months and we analyzed the adults. So 2 days of exposure, 6-month period, metabolomics and lipidomics analysis. And what we found, strikingly, was that there were hundreds of metabolites and lipids that were massively dysregulated. And some of the evidence here, I'm not going to go through it. I could do a whole talk on this. But for example, we see tissues that don't normally do beta oxidation that start doing beta oxidation. These are the kinds of dramatic metabolic phenotypic alterations that we're observing. So if anybody's familiar with the developmental origins of health and disease, I think this underscores that sort of concept, that hypothesis, that it really could be true that early exposures could lead to massive alterations in metabolic dysfunction later in life. But to study this, as I said, we need data from early on. So how do we get data samples collected, when you're an infant, to study someone who has Alzheimer's when they're elderly? That's the sort of thing that we want to think about. Now, when we think about multi-omics and we think about scaling this, because as I said, the argument that I want to make today, really the centerpiece of this talk, is that for multi-omics to deliver on our goals of precision medicine, we need those 3 things: big, diverse, longitudinal cohorts. And that means lots of samples. And I've taken the omes, the multi-omics, I've divided them into 2 buckets. On the left, I have whole genome sequencing, bisulfite sequencing, ATAC-seq, and RNA-seq, and I'm going to annotate those as sequencing technologies. And the reason I've done that is because we've really, is a research community, figured out how to scale those up, right? We've performed genomics on hundreds of millions of people. Something like a third of the population has been sequenced. So we really know how to do diversity, big, longitudinal cohorts with sequencing. The ohms on the right, however, are a different story. We've got proteins, metabolites, lipids, and chemical exposures. Unlike the sequencing technologies that are shown on the left, all of these ohms are profiled by using mass spectrometry. I'm going to put a little asterisk there and a caveat. You could, if you wanted to, you could move proteomics to the sequencing technologies because there are new approaches like Olink and SomaLogic that allow you to do proteomics with sequencing technologies. I would argue that we still at least want to consider proteomics as a mass spectrometry science because there's a lot of information that we can get from mass spectrometry that is unknowable by using the assays from SomaLogic and Olink. But, you know, acknowledging that it could really appear on both lists. So the question is, and what I'm going to spend a lot of time focusing on, is how do we get the technologies, the omic layers on the right, how do we move them over into the same category as the left so that we can deploy them at a population scale that's analogous to sequencing? Initially, you might say, maybe it's a sample prep and an instrumentation issue. But actually, that is not true. Mass spectrometry is readily scalable. In fact, newborn screening is used for almost all babies that are born in the United States go through some type of screening with mass spectrometry for inborn errors of metabolism. Now, that's different from what we're talking about today. Those targeted analysis for, you know, a couple hundred compounds that are indicative of inborn errors of metabolism, so not discovery, what we want to do. But it does demonstrate that mass spectrometry is scalable to the types of numbers that we're talking about here. And we have a lot of robots. And mass spectrometers, we can automate. You know, we run tens of thousands, hundreds of thousands of samples a year. But where the challenge is in data processing. That's really the complication. And the reason is shown here. So what I'm showing you on this slide is the exact same analyte that was analyzed over and over. Is a very boring set of experiments. The same analyte analyzed over and over and over again, 2,000 times. And the y-axis is its signal intensity. It's the exact same analyte, same mixture, same sample. So what you would really hope for is an absolutely perfect flat line, right? The exact same signal intensity, the whole duration of the 2,000 runs. You can see clearly that's not what's occurring. The mass spectrometer is very sensitive to temperature, humidity, seasons, all these sorts of things. The amplifier voltages change. And so you get these drifts. Okay, and that's really the challenge and what we're going to talk about today for overcoming. Okay, so that's really the backdrop of what we're dealing with. And what I'd like to do is talk with you today about some of the advances that we've made to make mass spectrometry scalable. I'm specifically going to focus on metabolomics, lipidomics, and exposomics. But we've got some data for proteomics too. I just don't have time to share it with you today. So before I get started on this section, what I'd like to do is say a few words about nomenclature. What do we call, if you're profiling metabolites, exposures, and lipids, I could say metabolomics, exposomics, and lipidomics every time. But instead what I'm going to do is I'm going to lump all of the discovery analyses of small molecules. I'm going to call all of that together non-targeted analysis. And I'll put that on the right side. And this is really our perspective of the molecular universe. We have molecules that are small, which are exposures, metabolites, and lipids. And when we do discovery profiling on that, we're going to call that non-targeted analysis. And everything on the other side of the screen are the macromolecules. Those are going to be the big things: RNA, DNA, proteins, all that kind of stuff. Okay, so what I'm going to spend this whole section and actually the rest of the talk discussing is the application of non-targeted analysis to large numbers. So metabolomics, lipidomics, and exposomics. Okay, so I've already shared with you that when you run samples over and over, we have a drift problem. We've known about this forever. Okay, when we first start doing metabolomics back in the early 2000s, we recognize this is a problem. And look at that N. That is a terribly underpowered, weak, pathetic N of 10. Right? But if you run, even if you're just doing 10 samples, each dot there represents a different peak. Sometimes in metabolomics or lipidomics or exposomics, we call that a feature. Each dot there represents a different peak or a signal or feature. And when you just take something like a blood sample, in our lab, when we do LCMS, liquid chromatography, mass spectrometry on that, we're measuring about 25,000 signals from blood. And you can see that if you compare two samples, the amount of drift that you're observing is considerable. And the y-axis there is the time course of the experiment, and the x-axis is the time course of the experiment, and the y-axis is the intensity. And you can see that the drift is all over the place. It's not linear. You can't just say, well, it's always 20% higher. It goes up, and then it goes down, and then it goes back up, and then it goes down. It's all over the place. Now, fortunately, back in 2005, we developed a software platform called XCMS that addresses this problem. It is designed to solve this, and it really enabled, at the time, untargeted metabolomics, non-targeted analyses. This, as I said, was back in 2005. There's a lot of other software out there. It's certainly not that XCMS is the only one. There's MS-Dial, there's MZMine, there's MetaboAnalyst. There's actually over 300 different softwares available now to do this kind of analysis. I'm just showing you XCMS because that was the one that's one that I've been involved in. But whether it's XCMS or MS-Dial or whatever your favorite software is, if you take it and you apply it to a large number of samples, this is actually just 2,000, and I say the word just because that's much smaller than the hundreds of millions that I've argued that we need, but it's bigger than what we did in 2005, which was 10. You see that you're really kind of up a creek here. In 2009 files, you're looking at 1,100 hours for this analysis, this data processing to go to completion. And actually, I regret to tell you that it won't even finish. This program will crash before you get a result. So this is an unusable platform. And as I said, maybe we're making progress. I was at a conference a couple weeks ago. I know that Shusha Du at UNC Charlotte is doing some beautiful work to scale up. So I think we're making progress here. But in general, this is what we've got. And that includes the commercial platforms, by the way. So if you're using the commercial software, it's the same issue. So what can we do about it? Well, in 2014, we published this paper, which I'm not going to go through in great depth, but I'm just going to briefly mention it, that demonstrated that, I mentioned earlier that when we do non-targeted analysis, that you're detecting on average about 25,000 peaks. Okay, but most of those peaks are not biologically insightful. Okay, more than 90% of the peaks that you measure in an LCMS-based non-targeted analysis, whether it be metabolomics, exposomics, lipidomics, the majority of those peaks are not physiologically relevant. Okay, we developed this technology in 2014 called credentialing that we used to first make this observation, and it involves stable isotopes and a bunch of different experimental idiosyncrasies that I'm not going to go through today, but I'd be happy. This is obviously a passion of mine. I'd be happy to chat with you about it online. It's been replicated by a number of labs, a lot of different results, all converging on the same notion that the majority of peaks that we see in these data sets are, in fact, noise. Okay, let's take a little bit of a deeper dive into the composition of these data sets. I got one slide on this, but it's animated. And again, it's a little high level, but I do want to paint this because I think if you take one thing away from the talk today, it might be this, that these data sets are really complex. And if you don't filter them and you don't clean them up, I just worry that a lot of what you're measuring is not going to be insightful. Okay, so let's think about what is the nature of the beast. 25,000 signals. Of them, some of the signals are going to be biological and some are not. Now, your instinct might be to say that non-biological, that's where the exposures are, but that's actually not what I'm meaning to communicate here. Non-biological, what I mean here is that these are signals that do not originate from the sample. I'm including exposures in the biological because those come from the sample. I mean non-biological, I mean two things, contaminants and artifacts. These are not contaminants to you as a human. I know this gets confusing because we're transversing fields. These are contaminants that are introduced during sample analysis on the analytical side. So what is an example of a contaminant? Well, if the graduate student preparing the sample sneezes into the vial, right? That would be an example of contamination. Or if you're using plasticizer, if you're using vials, plastic tubes, the plasticizers get leached into your sample. That's a contamination. If you have carryover from a prior experiment, you're using a dirty column and there are still molecules residing from the prior experiment that show up. That's an example of contamination. We also have artifacts. Some people use the word contaminants and artifacts synonymously. I don't like to do that. I like to distinguish them because I think they are different kinds of signals. Artifacts, they're not real molecules. You see a signal, the software tells you there's a signal, but it has nothing to do with anything molecular. It is, in some sense, it's because we're greedy. When you do an analysis, we want to detect as low concentration things as we can. And when you look at the baseline, it's noisy. And sometimes the baseline just deviates a little up and you go, that's it, that's a PFAS, right? Or whatever your favorite chemical is. But in many cases, it's not. It's just a deviation of the baseline. But because that could look like a signal, and we're greedy, the software will call it a peak, a signal. But that's an artifact. We also have, of those biological signals, those come in different flavors. We have those biological signals that are unique and signals that are redundant. What I mean by redundancy is that if you take a chemical, and we have many examples of this that we've published, there could be one chemical that shows up as 200 peaks in your sample. Okay, 200 distinct peaks with different MORZ values. Why? Well, this is some of the reasons. Each chemical can form salts with different adducts with different salts, sodium, potassium, lithium, whatever you name it. There's naturally occurring isotopes, which I know some people in this room enjoy looking at. And they can provide a lot of insight, but they do cause more peaks. Those are oligomers. Chemicals can dimerize or trimerize or whatever with each other, or oligomerize. And also there's fragments. So a single molecule, when it, glucose, when it goes into the mass spectrometer, could break into smaller pieces. So that causes a lot of degeneracy, as we call it. Then of those molecules that are unique, there are those that we can identify, those that are known. Those are going to be your amino acids, your fatty acids, TCA cycle metabolites, glycolytic intermediates, et cetera, et cetera. And then there are those that we can't identify, which I'm referring to here as novel chemicals. In our lab, we've annotated literally thousands of data sets. And the relative distribution of these signals is shown here. So in our experience, it's actually something like 97% of the signals are not biologically useful and need to be discarded, need to be thrown away. And if you're doing multi-omics and you try to take these signals and try to integrate them with other omics layers, you're going to get complete garbage because it has nothing to do with reality. And you're projecting really bad data onto otherwise good data and trying to make inferences. So I really believe for multi-omics, it's critical that these get removed. Now you'll notice that the fraction of signals that are unique are really small, right? 3%-ish. Some are known, some are novel. You might be tempted to say, that novel, let's just get rid of that stuff. Who cares? Look at how it's small. But I really just want to take a moment and emphasize that you should not do that if you can, because there is a lot of really juicy stuff in that novel component. These are just a couple of the examples, a few examples from recent years that we and others have discovered. These top 3 molecules here that contain sulfur. We recently reported these from Gut Microbe. Dimethylsphingosine is an interesting example. It's sphingosine with 2 methyl groups. We found that about, 10, 15 years ago now. That's actually present in the CNS only, and it mediates some pain responses. This lactate phenylalanine is a really interesting compound. This came from Stanford. It's a product of the gut microbiome, and it's hugely important in regulating physiology. It's showing up in all kinds of things. I think that's got it. We're just beginning to scratch the surface with that one. And then on the right, you've got all these bile acids that are emerging. There are literally hundreds of new bile acids. So you take whatever your favorite bile acid is, and you attach an amino acid to it or a glycolate, whatever. You can add all these things to it. Somebody who studies bile acids told me that when they were in medical school, they learned that there was 10 bile acids and now there's hundreds because we've expanded our repository so much by doing these characterizations. And these are all really important. So you don't want to lose that stuff. Discovery is still important. I don't want to discourage you from doing discovery because this is why. Now, if we're going to take mass spectrometry and we're going to reach the level of sequencing, my opinion is that we need to do this, what's shown on this slide. If you try to do data processing with all 25,000 signals, I showed you what happens. 1,100 hours in a crash program. That's what you end up with, and an unhappy postdoc or graduate student. On the right is the situation if we can get rid of the stuff that isn't important, and we can data process off that, things go a lot better. And that's the strategy that we've assumed. I'm not saying that we can never figure out how to do the 25,000, but I don't want to be a victim perfection of, paralysis by perfection. I think that we can go off and do this right now by filtering. So let me tell you exactly how we do that. This is somewhat high level, but I hope that it will be useful to you. And again, I'm happy to chat more about this offline. I know there's varying degrees of expertise here. Some people are in the weeds with the instrument, and then other people are more high level. So I'm trying to accommodate everyone. But like I said, please let's talk after if you want to know more. Okay, so this is how we do it. We take a small aliquot of the samples in our cohort, and we pool them. We make pooled samples. Okay, the question you're going to ask me is, how many pooled samples do you need? That's $1 million question. Our goal is to use 2 orders of magnitude fewer pooled samples than total samples in the cohort. 2 to 3 orders of magnitude. You don't want to have having more pooled samples is great, but if you have n-1 pooled samples compared to regular samples, you're not saving any time. So the study I'm going to talk about, which is 20,000 samples, we had on the order of about 200 pooled samples. We take those 200 pooled samples and we do complete annotation on them. So I just told you on the previous slide, I showed you the big plot of all the different kinds of signals. We do that for those 200 samples. We annotate them completely in totality. And when I say annotate, you don't have to figure out what they are. You don't have to identify every novel compound, but you want to know that it's a novel compound and it's biologically relevant. That's what I mean by annotate. I don't mean like level 1 identifications. I mean, let's just figure out what we care about and what we don't care about. Then we collect the data. So all the data is there. That's a really important point. We don't do anything to data process. All the data are there. As I said, the actual data collection, that's not a problem. That's not limiting. So collect all the data. And then when we do the data processing, however, we only focus on the signals that we pulled out that we deem to be biologically relevant. We take all the signals from the 200 pooled samples, and that's the only part of the data set that we process. Plus all of the signals from our database. We have a library, internal library, with about 6,000 compounds that we have, and we also screen for those, about which of 3,000 are environmental chemicals. Okay, so that's great. That helps with data processing, but it does not solve one of the major issues that I pointed out at the beginning, and that is you get this variance in signal. Okay, and that is to say, this is one of my favorite compounds, LPC 16-0. You'll see why I say that in a couple minutes. This is the intensity, and this is its intensity as a function of run order. And you can see that it's all over the place. And your question is, okay, I can process it, but that signal's all over the place. Is that because of analytical variability, or is that because something interesting is happening biologically? And we also solve this with pooled samples. So we run not all 200 pooled samples, but one of the pooled samples. We pick one of them, and we run it every 10 samples. Every time we run 10 research samples, we run the pooled sample. And then what we do is we use that to calibrate. That should not change. Any change that you see in the pooled sample is because of instrument drift. And so what we use is a package of 14 different machine learning algorithms to correct for that. What I'm plotting here on the y-axis is how well they work. It's minimizing analytical variability and maximizing biological variability. And each one of these is a different approach that we use and test. You might say, well, okay, clearly random forest is the best over there on the right. That's certainly the case here. But for reasons that are unclear to me, it varies from project to project. Sometimes random forest doesn't win and a different test is used. So we check every single study we do, we test it. So what happens? Well, this is, again, around, I think this is around 2,500 samples. You can see before we do correction, the batches are color-coded. And you can see, actually, I guess this is about 1,000 samples because it's about 100 samples per batch. And you can see that the samples are color-corrected. The batches are color-coded. And there's clear separation, which is, of course, bad. But after we do the correction, then you end up with a situation that looks like this. So that's good news. Okay, so what I'd like to do is tell you, I've told you all of this. That sounds great, but the proof is in the pudding. So let's talk about what this actually looks like in a real application. And the application that I've chosen to share with you today is a project that we've been working on in collaboration with a number of my colleagues at WashU. The leader on this project is Michael Province, but also in collaboration with NIA. This has been a study that's been ongoing for several decades, called the Long Life Family Study. The goal of this study is to understand healthy aging. The NIA developed families, several hundred families, that generation after generation after generation live to be over 100 years old. So knowing that these folks were probably going to live to be over 100, they insightfully started to collect samples earlier in their life, so that they could try to get, because often when you do aging studies, you take the blood from a 105-year-old and you compare it to the blood of a 40-year-old, but it's not the same person, right? We were talking about that. It's really hard because, like, you know, if we collected blood from children, we'd all be dead by the time that they got to the age that we wanted to study them. But the samples existed, so it was really insightful, and we're lucky to have those samples. So far, we've done non-targeted analysis on 12,000 plasma samples. We've got 8,000 more that we're doing now, and that number is probably bigger than that now because this slide's a little old. Okay, so let's talk a little bit about what we found. We found 308 chemicals that predict age. The p-values on the y-axis, the fold change is on the x-axis there. This is a volcano plot. So this is the amount that they change per year of life. There's some usual suspects on there, but there were some chemicals that we were surprised to see. I could spend a whole lot of time talking about this, but what I'm going to talk about are really two things that I think are among the most interesting that came out of this cohort. Because we're tracking these patients, we're also doing other omics, by the way. I'm not talking about it here because it doesn't fit in. I know this is a multi-omics idea, but we've got genomics, we've got, we're doing proteomics, so we've got other data here too, but I'm only going to focus on the non-target analysis here. But in addition to that stuff, we also have metadata, so we know when these patients die. And so what we can do is we can actually build survival probability curves. We can look for chemicals, metabolites, lipids, and exposures that go up or that plummet before death, and ask, if this metabolite goes up, does that mean that you're going to die? Essentially, that's the analysis we could do. And I'm showing you one here. It's a polyunsaturated fatty acid. And I've divided the cohort here, and it's a little hard to see, but hopefully you can see it. Into young, which I'm going to call less than 90, which always makes everybody feel good. So if you're less than 90 in this cohort, you're young. If you're over 90, you're old. Okay, and you can see that those that are less than 90 have a higher survival probability than those that are over 90. Obviously, there's a bigger chance you're going to die if you're over 90 than if you're under 90. But you can see that if this polyunsaturated fatty acid is high, that top line, you have a higher survival probability than if it gets low. So if you're less than 90 and the polyunsaturated fatty acid is high, better chance of survival. If it starts to drop, chances of survival go down. And that same pattern holds, by the way, if you're over 90. Okay, it's even perhaps more pronounced. Now, the polyunsaturated fatty acid, we've known, there's a lot of evidence out there that this polyunsaturated fatty acid is important to health, and that its deficiency can be detrimental to human health. So in some sense, this is a little expected. But we wanted to ask if there's anything else maybe that we would not have predicted. And I'm going to share with you one of the environmental chemicals I think is really interesting, a dietary environmental chemical, tartaric acid. So its relative level is on the y-axis. The age of the subjects is on the x-axis. We have 12,000 points there. And you can see that it generally goes from low to high as you age. And we can build the same sort of survival probability curves that I showed you earlier. And this one is rather striking. You can see if you're less than 90, it's the top two lines. If you're older than 90, the bottom two lines. And irrespective of what age you are, if your levels of tartaric acid get high, it's very bad news for your future. Your chances of surviving drop by half. So if you're less than 90 and tartaric acid goes high, there's a 50% chance you're going to die. If you're over 90 and your tartaric acid is high, you have very little chances of surviving. Now, I want to be careful. It's not to say that this is causative, right? This is obviously a correlative relationship. But it does make you ask, where is tartaric? Where do we get this? I said it was a dietary chemical. It is. There has generally been a perception that tartaric acid is good for you, and they've fortified all sorts of drinks with it. Does anybody drink kumbaka? Yeah. So they sell this at Whole Foods. It's very popular, and it flies off the shelves, and it has very high levels of tartaric acid. Whenever I fly, I love to enjoy. I don't know why. I've been doing this for years. I like to buy orange Tic Tacs. If I'm flying, I've got orange Tic Tacs in my suit pocket. And I discovered on my last trip that the second highest ingredient in orange Tic Tacs is tartaric acid. So it's all over the place. Now, we don't think that when you're old, you start drinking more kumbaka, and then you die, right? That's not, we don't think that. But there is an interesting question why this happens. And we're doing some mechanistic studies now to try to understand that. And what we think happens is that tartaric acid is primarily metabolized by the gut microbiome. And so what we think this likely reflects gut dysbiosis, and that you've got some sort of breakdown, and maybe that it's the gut microbiome that's giving you this signature. But nonetheless, I think this reflects the kind of interesting things that we can get out of these population studies. Okay, and some of this data has been published recently. Okay, I'd like to, in the few minutes that remain, tell you a story about dietary fructose and cancer. I'm going to talk about fructose because fructose is among the most prevalent dietary components that we're exposed to. Some people may try to avoid fructose, and you're a better person than I am. It's obviously very addictive, at least to me. I love sugar. And if you like sugar, it's almost impossible to avoid fructose because it comes in the flavor of high fructose corn syrup. And it's definitely been one of these things that has changed considerably over the last 100 years. So in 1922, the average person in the United States consumed 7 pounds of fructose, which is about equivalent to the weight of one gallon milk jug. In 2023, 100 years later, the average person consumes the weight of 15 1-gallon milk jugs. So it's gone up considerably. There's a lot of reasons for this, in part, much as everything in the exposure world, driven by economics. High fructose corn syrup is sweeter and it's cheaper to make and use. And that's one of the reasons why it's been used so extensively. Okay, so who cares? Well, fructose is fructose has the exact same molecular formula as glucose. They're structural isomers. The difference is that fructose enters into glycolysis beyond the regulated step. So one of the reasons that fructose is thought to have contributed to the obesity epidemic is because the more you eat, the more you metabolize. There's no off switch like glucose. So it's been pretty well appreciated how that contributes to obesity, but we were interested in how it might affect tumors. Tumors we know are addicted to glucose. This is a PET scan. About 1 million people a year in the United States, we infuse with a radioactive version of glucose because tumor cancers are so addicted to glucose that they take it up faster than any other cell. So this poor person has tumors in their liver, and that's what you're seeing here. So we wondered if all this fructose was detrimental to people who have cancer. And so what we did is we implanted tumors in mice, and then we gave them a diet that was normal, standard diet, or we gave them a diet that was enriched with fructose. And we monitored the tumor progression. We asked how the tumor evolved with time. And you can see in every scenario, we looked at all kinds of cancers. This is just a smattering of what we evaluated: cervical, melanoma, breast, lung, all kinds of different cancers. Cervical is on there twice because we did multiple types of cancers, even of the same type. And you can see in all scenarios, unequivocally, fructose drove increased growth of tumors. Really scary. In some cases, like 4-fold. When you think about how fructose is metabolized, it's the multi-omics component of this project. When you think about how fructose is metabolized, it makes fructose 1-phosphate, and that feeds into glycolysis, as I said. There are 2 enzymes that are important to making that happen: KHKC and aldolase B. So we knew that fructose was driving growth of these tumors. When we looked at the tumors, we expected these 2 enzymes to be present. And we were surprised when we discovered that they were not. KHKA, which does not readily metabolize fructose, was present, but KHKC was not present. These are human tumors. Aldolase A was present, which is used for glucose metabolism. Aldolase B was missing. And we saw the exact same thing at the gene level. Sorry, I showed you gene expression. This is protein levels. So the proteins are just missing. And furthermore, if you take cells, and this is again just eight cell lines, and you take these cancer cells and you grow them in media that has glucose, media that has fructose, or media that has no sugar, which is C, the cancer cells don't proliferate if fructose is present. They basically, it's like having no sugar present. They cannot use fructose. The cancer cells cannot use fructose. So this led to this pretty puzzling question. Fructose drives tumor growth, makes the tumors big, sometimes 4 times as fast, but they can't actually use fructose. So how in the world does that work? How's that happening? So this is the model that we came up with. Fructose is consumed by the patient, but the cancer cells cannot utilize it. Instead, what happens is the fructose goes to the liver. It gets metabolized by the liver, which, by the way, I'm not showing you that it does express the fructose metabolizing enzymes. The liver converts it into another nutrient. The nutrient gets excreted by the liver, travels back to the tumor cells take it up, and that drives growth. This is really cool, but sort of like looking for a needle on a haystack. Because how are we ever going to figure this out? The nutrient that gets excreted in the blood is likely a nutrient that's present from all sorts of sources. And we really want to know, does the nutrient come from fructose? And does it come from the liver? And how do you track that? So the way that we've been doing those experiments is by using stable isotope tracers. These are not radioactive tracers, which is what we use when we do PET scan. These are stable isotope tracers. And we can infuse them into animals like mice, and we can also infuse them into people. So what we do is we hook up an IV line about 3 hours prior to resecting a tumor. We infuse with isotopically labeled nutrients. Then we can look at the tumor and learn what's going on with the individual. Now, it's kind of an analogy to explain why this is useful. I'm gonna use this this toy example. I packed up, went to the airport, put everything in my red suitcase here, and it looked great, right? It was at home. Everything I knew was exactly my suitcase. I dropped it off at the check-in desk, got on my plane, flew to LaGuardia, went to LaGuardia Airport to pick up my bag, and you're up with a situation that looks like this, right? Where there's 100 other bags. This is obviously hyperbolic, but there's 100 other bags that looks just like this. Okay, and how do I know which bag is mine? This is the same situation with fructose. Fructose comes out, makes a nutrient. The nutrient goes into the blood, but there's a million other nutrients from all other tissues, all kinds of other precursors. And how do we figure out which one is derived from fructose? So the way we solve this problem when we travel is we use luggage tags. All right, so who's got the brightest scarf or whatever it is, sticker or bumper sticker or whatever that you tie to your bag. And so you could do that, and then you know when you get to baggage pickup exactly precisely which bag is yours. So that's what isotopes do. We label fructose, then whatever is derived from it has the luggage tag on it, essentially, and we know where they came from, you know, that it's where its origin, its provenance. Now, currently in multi-omics studies, this isn't really tractable because, as I said, the key to multi-omics precision medicine is large sample numbers, and doing isotope tracers, like when we do that in the mice, it takes forever. It's expensive, and so. But what we've been doing recently is using deuterated water. And we do this, I showed you earlier with stable isotopes, we do that in the clinic. You need a team of nurses, you have to test it's a lot of paperwork. It's really a headache. For example, we have a clinical trial where we're using isotope labeling to make clinical decisions. But with water, it's really great. It's deuterated water, it's cheap. And you can do it at home. So we give the patients a bunch of water. They take them home. For the first 5 days, they take 4 a day. It's just a little vial. It's not even this big. It's very small. And then for the next about 4 weeks, they take it twice a day. We collect all of the used vials. When they come back to the program coordinator, they have to deliver the vials. So we have some sort of compliance report. And we get beautiful labeling. It works just as well as when we're doing the more complicated situation. I believe this is the part of the future of multi-omics, is doing these sorts of studies with large scale using deuterated water. It's not yet, like I said, it's not yet in widespread use, but I really hope that'll change and we can start to build this into multi-omic studies. So by doing that, what we discovered here, and I know I'm running a little short on time, but I'll get through this, is that fructose makes a type of lipid called lysophosphatidylcholines. And those lysophosphatidylcholines get excreted from the liver, enter the circulation, and then get taken up with enormous avidity by cancer cells. Cancer cells absolutely love LPCs, lysophosphatidylcholines. If you look at an animal that's on a high fructose diet compared to an animal that isn't, the most dysregulated thing that you see in the serum is LPCs. LPCs, as you can see on the right there, that plot, high fructose corn animals on high fructose corn syrup have, this is just one LPC, 18-1, is way higher when you're on high fructose corn syrup diet compared to controls. Okay, and that's just one LPC. They're all following similar trends. Okay, so LPCs go up, which makes sense. If it's going to drive tumor growth, you'd like to see it go up in the serum. And we know if you take cells in culture or tumors and you give them LPCs, that it drives growth. So on the left, this is one cell line, but we've done this with, I think, probably 20 cell lines. On the left, this is TC1 cells. If you give them LPCs, they proliferate, whatever that is, 5 times faster than if they don't have LPCs in the media. And in vivo, if you implant a tumor and you IP infuse LPCs, the tumor grows much, much faster, as you can see, compared to if you don't infuse LPCs. Okay, so what we have here is that fructose makes LPCs, and those what's feeding and driving the tumor growth. Now, of course, this raises the question, can we block the metabolism of fructose in the liver? And if we do that, is that sufficient to prevent this effect on tumors? And the answer is yes. So this is, again, we implant tumors. We give them either high fructose diets on the top, standard diets in the middle, or a high fructose diet plus this drug, which is an inhibitor of fructose metabolism in the liver. And you can see that it really mitigates tumor progression. In fact, the animals do even better with this drug than they do on standard diets. And there's a reason for that. I won't get into it today, but it's because actually you make some endogenous fructose without even consuming fructose in your body. So we think it's a really good, possible, attractive solution. And actually, this paper just came out, or is about to come out in the next couple of days. And I just gave you a sampling of it. This is actually the first time I've talked about it. But if you want to know more about it, happy to read. Feel free to reach out, or otherwise take a look in a couple of days when it comes out. Okay, so I just want to make some concluding remarks here in the last section, and that is, how does multi-omics, all these studies, large scale, how is this informing how we think about disease? How is it redefining our perspective? So, and I'd like to leave you with these thoughts. Organisms are comprised, obviously, of many, many, many cells. And those cells, although we think metabolically, we tend to think of them operating autonomously. When you take undergraduate or medical school biochemistry, you see glycolysis and you see the TC. It's all contained and nightly. It's complicated, but all nicely organized. But that's really not what's happening. All of the cells are coordinating with each other. The output of one cell is the input of another cell. They're all connected. Okay? And so when there's a dysregulation, or one of those cells or some region of cells becomes pathological, it's not limited to just that tissue. That disease pathology, because of these metabolic couplings, gets propagated throughout the entire system. Okay, this is very relevant to exposures, because often when we think about exposures, we tend to think about the cells that the exposures is coming in direct contact with. So, lung cancer, or lungs if you're smoking, that sort of thing. But I think that's a very narrow view that we need to move past. In this case, what we have, I showed you that we have, this is a little different, and this is a lung tumor. And all of that green is metabolic dysregulation that's present across the animal. Okay, when a patient or an animal has a tumor, we've known for 100 years that the tumor has really wonky metabolism. But the other cells across the entire system, across the entire patient, have messed up metabolism too. It's not just the tumor. Okay, and that's useful because what it means is that if we can target other healthy tissues like the liver, we can actually interfere with disease process. You don't have to go after the pathological tissue. You don't have to limit ourselves to just the tissue, the disease tissue. In this case, I showed you an example where we're targeting the liver. The liver had no tumors. We were talking about cervical tumors or breast cancer tumors, and we developed a drug that goes after the liver. And by influencing the metabolism in the liver, it was able to mitigate tumor growth elsewhere. Okay, so I think that's a really important point. And we really need to move beyond tissue-specific, tissue-centric perspectives of disease. You know, if you have lung cancer, yes, it is about the lung, but there's dysregulation across. We shouldn't just think of it as a lung disease. If you have Alzheimer's, it's not just a brain disease. Okay, it influences, everything is connected. It really, you will find metabolic dysfunction in every phenotype and every disease we look at. Metabolic dysfunction spans the entire population of cells present across the system. We need to pursue large longitudinal cohorts. I've talked about that. I've emphasized that throughout. And I think we need to perform multi-omics profiling, not just on the disease type like the brain and Alzheimer's, but everything, including the blood and other fluids. So with that, I wrap up here and thank the people in the lab who were responsible for this work. Darshak did the exposure experiment with BPF that I showed you earlier. Kevin, Ethan, Mickey, Miriam, Nate, Cassandra, they've all contributed to the development of the platform and that middle part in the LFS long life study. And most importantly, Ronnie over there on the side, he led the wearing the fructose shirt and eating the fructose cake, both symbolically and realistically, is Ronnie, and he led all that work. Thank you for your time, and if we have any time for questions, I'd be happy to entertain them.
I'll start. So first, I lived in Atlanta during that Super Bowl. Thanks for reminding me why I've never watched.
Oh yeah, gosh, see if I was going to Atlanta, I would have deleted that slide. But yeah.
So I think I have a sense of your answer to this, but you know, in talking about that systems level effect, like the organ specific things are great, and you know, we have extracellular vesicles coming through the organs. But I mean, your view though is that blood or plasma is a good starting point.
Absolutely. And I will admit that is a big change in my thinking. So 20 years ago, when we started doing these analyses, I thought blood was terrible. I hated analyzing blood because I thought it's not, I want to look at the disease tissue. Like, what am I going to learn by looking at blood? It's secondary. But actually, I now believe blood is among the most important things that you can look at because all the metabolic networking and communication, that all happens through the blood. And the stuff that's interesting, you start thinking about therapeutic targets, it's all in the blood. I really believe blood is among the most important matrices we can look at because it's what connects every tissue. And diseases are not one tissues. It's a holistic. Diseases are holistic. You don't just have one pathological tissue. You die from cancer not because of the tumor. You die from cachexia usually, which is not So I think blood is. We should be looking at blood more.
I think this is like for precision medicine. Like we think about, you know, there's been consortia looking at the target tissues to kind of show that there's something there.
Yeah.
But blood tests have been a staple of medicine forever.
Yeah, absolutely.
The idea, there's just so much more information
there
in your CBC. So and I think that's a place where,
especially if you do multi-omics. Yeah, I totally agree. I think that's a great point, and I completely second that.
In the one, the sort of serum for the 90-year-old, one of the hit lists is tryptophan and caroline. Is that related to NAD and aging?
What was the hit?
Tryptophan.
Oh, tryptophan. Is it related to NAD?
Is it related to the NAD?
Like sirtuins and that sort of pathway?
Yeah.
I don't know. We haven't dug into that.
But does pick up on the blood.
Yeah. In the blood. Yeah. Yeah. I mean, we have a study on NAD from this cohort. I can send it to you as in Science a couple of years ago. But I don't know if that, I don't know. So I think there is an NAD component, but I don't know if the tryptophan is connected to the NAD. I know it could be, but I don't know if that change is related. Yeah. It's a good question though. Yeah.
That slide that you showed that 95% or maybe even more of the signals in your mass spec spectra, where the peaks are contaminants or artifacts.
Yes.
Is there overlap between contaminants and real biologically relevant signals that we're just losing that data because of the presence of the contaminants? I mean, I think the example that you mentioned is that grad student sneezes. Well, that sneeze it's going to carry some of the signals that are relevant, right? And it's going to,
Yeah.
So if they're just floating around, I don't know, like dander and other contaminants from people that are ending up in these samples, they could be masking
Yeah,
and adding like offsets that are
Yeah.
variable from day to day.
yes.
So is there ways to kind of address that? Because it seems like it could be a problem where we might be losing a large fraction of the biological signal because of all the other stuff that's not well controlled.
It's a fantastic question. And my advice would be don't sneeze in the sample. But yeah, no, it's a great question. So what we do is every time we make 96 samples, we have 3 blank samples that contain no biological specimen, that just has the water, organic, and it goes through every step of our process, and it sits in the middle of all of the other samples. And so whatever torment the other samples go through, the blanks also go through. And then we use those blanks as a readout of what might be coming from if you see something in the blank that's present in your sample, then you know that's potentially a major problem. The general threshold that we use is, so you could try to say, okay, well, it's in the blank at this level, I'm going to subtract this much off of my signal. We have not been successful at doing that. So one of the examples of what you're talking about is, does anybody bake cupcakes? Yeah. So, I don't. I'm terrible at cooking. But when you have like a pan with all the holes in it and you pour the batter and then you put it in the oven and then you take the cupcakes out. And if you're like me, when you take it out, you pull the top off and the rest of the cupcake is stuck to the plate. But you spray your pan with like, what is it called? Spam or whatever, some sort of oil, right? So that it comes right out. So when they make Eppendorf tubes, they do the exact same thing. They have molds and they spray them down with lipids, and then the molds, that's how they pull them off so they don't get stuck on them. So that all of your tubes are contaminated with lipids. So you need to know like which batch, so I encourage everyone that's doing these sorts of experiments to characterize their tubes, and figure out what the signals are. There are ways to get rid of them. So we buy tubes and we have a washing protocol that we use for all of our tubes. We also use 99, when we first did this, my students would come in and say, we can buy acetonitrile for $40. And they say, well, I want to buy the acetonitrile that's $2,000. And I'm thinking, it's $40 for a liter and you want $2,000 for a liter. Like, how important is 95 versus 99.999? It turns out it's really important. I would spend another $10,000 on solvents if I could buy higher purity solvents, because that is a huge part. So we get as high purity solvents as we can. And then we try to wash all of our vials and all of our tubes and everything to remove that. We've developed protocols to minimize. So you can imagine, once you've got your samples, you can run them and say, okay, I've got this much background. Let's try this cleaning protocol. And we've actually worked in the past, years ago, we worked with Eppendorf to try to develop, you have DNA grade tubes that you buy specifically for doing genomics, but why can't we have tubes that you specifically use for non-targeted analysis? And so we had that discussion about 5 years ago and there was no interest, but recently, in the last few months, that's reignited. And so I think there's a possibility that we could ultimately have tubes that would help with that sort of thing. The other thing is that once you know what they are, there are ways that you can infer what you're missing. So palmitate is an example of something that is incredibly important. It's the end product of de novo lipogenesis. It's absolutely critical to understanding fatty acid biosynthesis. However, it is unfortunately one of the SUP agents that's used in the tube, so you get a lot of background. But palmitate conjugates naturally with carnitine, so you make palmitoylcarnitine, and you can use palmitoylcarnitine as a proxy for palmitate. So when you know what your blind spots are, and then you can also rely on other omics data, you can start to fill in the gaps, but you have to be aware of it. That's the key thing. I know that was a long-winded answer, but yeah.
I guess I have 2 questions. So you mentioned the power of high-resolution spectrometers.
Yes.
For multiple omics. So what are the most important or essential differences between exposomics versus metabolomics? I guess you mentioned the sample collection, the tubes, you're probably part of it. So how can you differentiate the environmental exposure versus the metabolites? My other question following, the garbage as you talk about, so a lot of them are noises
Yeah.
in the signal. And you mentioned the importance of using filter to filter something out before we actually focus on multi-omics, that sort of analysis. So I wonder, because it's already challenging for metabolomics. How can you move this forward to exposomics, where the exposures are random, low abundant, and a lot of them are just noise level?
Yeah, no, it's a great point. Okay, so the first question is how important is high resolution? Is that basically what it was?
No, like the essential differences.
Oh, between exposomics and
Yeah.
metabolomics. I think that data processing is virtually identical. There is differences. The main difference is that, and we chatted a little about this at lunch. If we drew everybody's blood in here and analyzed it, everybody would have glucose. We'd all have, amino acids, right? That we'd all have that in common. But there would be certain PFAS or certain phthalates or certain acrylates that one of us might have and no one else has. So if you're analyzing 20,000 people, there could be samples that a chemical is only detected in one person, and that's important. And so in metabolomics right now, they use a detection frequency. And if that chemical is not detected in something like 75% of the samples, you discard it because it says, well, it must be an artifact. So I think one of the key differences is differentiating between what I would call one-hit wonders. These are chemicals that show up in one person out of 20,000, and knowing how to deal with those. Now, that is hard. That's the hard part. That's the gloomy part of the equation. Like, oh my God, what are we going to do? But the good news is if your signal only shows up in one person, it's binary, meaning that you don't really care. You don't have to deal with all the calibration, the drift that I told you where it goes up and down. That all goes away, right? Because it's absent, and it's present. So you don't really care about its quantitation, so to speak, because it's zero, it's affinity, right? The fold change is infinity because it's not present and it's present. So that part of correction is the good news, that we don't have to deal with that sort of batch effect aspect. But finding them among the data, that is the tricky part. And I think that I think there's a lot of potential. There's new instruments that are out now that are a lot faster. And I think that I could go on a soapbox and maybe recommend that we chat a little after. But I actually think that for exposomics, ultimately, we're going to redefine the workflow. And I think we're going to move. I think we're going to be, I hope in a few years, we're going to be doing very different kinds of experiments for exposures with leveraging new instruments. That was your first question. What was your second question? Oh, how do you filter, or what would you, or no, how do you filter? Yeah.
Bring this to where a lot of exposures are random and noise level already.
Well, I don't know. I don't know that filtering. That's a great question. We don't, I guess we've always assumed in metabolomics that we're thinking about chemical exposures. I mean, I think it gets kind of dicey when you start saying, well, metabolomics is different, but when we developed all these filtering techniques, we never just really designed it for endogenous chemicals. We're always thinking about exogenous chemicals. So you raise a good point. That's why it's hard. But I don't think that metabolomics, nothing we do in that filtering technique assumes that that's not true. So I don't know that it really impacts how we do that filtering. But you're right. Those are complications that make it difficult. Those are great questions. Yeah.
Just follow up on that. Do you have the luxury of having multiple serial samples from the same person?
Yeah.
Rare signal.
Yeah, but in our experiences, depending on how close those time points are, about the exposome changes about 50%. Like we've done projects where we've looked at the exposome over time, and if you sample the same person a year apart, their exposome is about 50% different. So that could help, but there's a lot of variance in the exposome.
Can you comment on so these technologies are still really expensive.
Yeah.
So can you comment on the trends in costs and what we can expect in the years to come? Because
Yeah.
one of the enabling thing about genomics was dropping cost of sequencing.
Yeah. No, it's a great question. And Gary and I were talking about that. There are ways that we could drop the cost. I personally don't think that I'm not an advocate of that because unlike genetics and genomics, where people kind of bought in de novo. I don't know that we have that buy-in for exposures and metabolites and lipids. And I'm worried that if we drop the cost and generate less quality data, that it may hinder enthusiasm. So I really worry about doing things cheap, less expensive, and sacrificing quality. But I think there are ways that we could get there eventually. There are ways right now, and if we talk about just the overall cost, if we look at whole genome bisulfite or whole genome sequencing data, right now on a per sample basis, this is still cheaper than that. So it is expensive, but it's not astronomically. It's not like it's 5 times as much as whole genome sequencing. I mean, I think it's at worst case, it's the same cost, but most likely half the cost or maybe even a quarter of the cost, depending. So it's not astronomically more expensive. I think that with new instruments and when we get faster, we can start to cut some corners. I really believe that the best answer to your question is that right now, everyone who does these experiments does them differently. The motto that we say, the joke that we tell in the field, and we all want to do them our way, and we don't want to do anybody else's way. It's like we say, it's like using someone else's toothbrush. Like we have our own stuff, and like this is how we do it. We wash our vials. I don't want to not wash my vials because I always wash my vials, and you don't wash your vials. You don't wash your, it's this weird. And so I think part of the challenge right now is that because we all do things differently, like when we do a cohort, we have to do all of it. We can't like split it with Gary because that would be, we'd have very different kinds of data. But I think if we could move, I think the answer to lowering the barrier for cost is to move to a harmonized method where everybody is doing things that are comparable, or figure out a way to harmonize it. Then we could take a large cohort and spread it out over a bunch of labs, and that would really substantially help with throughput, cost, and a lot of things. But we need, again, I think the first step is we have to convince everybody that this is an essential piece of the multi-omics puzzle. And once we get buy-in, I think we can start pushing some of those other buttons. I don't know if that answered your question.
Well, thank you very much. Really enjoyed that. If you have a question, we can follow up afterwards. Great.
Thank you.